You're listening to an Airwave Media Podcast.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We are your hosts, Dr. Jess Steier
and Dr. Sarah Scheinman.
And today we are going to tackle a topic that, honestly, I feel like I'm going to be doing a lot of question asking and listening to Dr. Scheinman. The topic is epigenetics. It's a really cool branch of science that I feel like, people hear the term but don't really understand what it means. It came up briefly in our episode on Alzheimer's. I can't remember the context. I don't know if you do, Sarah. Maybe you do.
Well, you were just talking to me about what my research was in particular and my interests, and what my postdoctoral research right now is focused on the epigenetics of Alzheimer's disease.
NBD, no big deal. And just to be totally honest, I remember, or I think I said something along the lines of, my understanding of epigenetics was that things in our environment are turning on certain genes, and you were like, you kind of jumped in, and we're like, well, that's not exactly what's happening. And so, really, I think today it's going to be time for school with Dr. Scheinman. But I also, I have lots of questions. We're going to talk about some landmark studies in epigenetics. And really, I guess the overarching conversation is about the interplay between nature and nurture, right? And so obviously we're born with a set of genes, and then we live in a world, and there's, the environment around us, and something's going on. There is some sort of interaction between those two things. So we're going to get into it. Anything else to set the stage before you sort of jump in with some definitions for us? We know you love a good definition.
I do. Well, I would say, we can't talk about epigenetics without talking about genetics, right? So let's just set the stage a little bit and just remind everyone some biology 101. When we're talking about the human genome, it's comprised of just 4 letters, right? A, T, C, and G. And these are, it's a very simple language when you think about it that our genome is comprised of. But those 4 letters are used over and over and over again. And this creates just an extraordinary volume of DNA. And you get 3,000 million of these letters from your mom and 3,000 million of these letters from your dad. And just one mistake in a single one of these letters can cause devastating disease. So your genome sequence is actually really important, and no one's debating that. Mm-hmm. But the thing of it is, and what we've come to realize over the past several decades in biology, is that that's not all that there is. And we've known this for a long time experimentally. And that's kind of where this, nature versus nurture debate originated that you were mentioning, Jess.
Right.
So that's what we mean when we talk about genetics, right? That blueprint, that raw genetic code that you're born with, that you get half from your mom, half from your dad.
My cat Marley just came to enjoy the podcast with us. Hi, Marley, if you're watching on video. So if you hear purring, that's what's going on. Okay, so here's my question. So, I've heard about the mapping of the human genome. It's so over my head. I can't even really grasp what that means. Have we mapped it? Is there more that we don't know? I feel like we're still learning a lot about genetics. Is that I don't know. Is that silly to ask?
No, it's not silly at all. Just because, the mapping of the human genome, and, we accomplished this back in 2003 and through something called the International Human Genome Sequencing Consortium. And more simply, you've probably heard of the Human Genome Project.
Right.
And that's kind of widely considered to be one of the greatest achievements in biomedical science to date. Because in sort of a little over a decade, scientists were collaboratively, and that's what's also amazing about it. There was such beautiful collaboration between scientists to make this happen.
Right.
We were able to sequence the 3 billion chemical base pairs, the A's, T's, C's, and G's, and map them to specific genes.
Incredible.
And I mean, it is really incredible. And I mean, like what we learned from that is, interestingly enough, is that only about like 2% of our genome actually codes for proteins. So there's a lot of genetic material that like we used to think of as what's called like junk DNA,
right?
But through through epigenetics and through these all these like new discoveries, is that like actually that junk DNA is actually doing a lot to regulate the expression of the actual protein coding regions of the DNA.
Got it. Well, so you're answering my question. It's like we've learned a lot, but it's not like the book is closed. Like obviously. Okay. So all right. No. So now we've talked a bit about genetics. So now what's epigenetics and how are these two things different?
All right. So epigenetics directly translates to like on top or above genetics. So when we talk about epigenetics, like this just represents information that's layered on top of our DNA that influences how the genes contained within the DNA operate. So this information does not at all change the blueprint, the genetic code. That is something that you are born with. You get, like I said, half from your mom, half from your dad. And that is something that you are born with and is just sort of like baked into every single one of your cells. What we've come to realize, though, through the study of epigenetics is that your genes do not define you. So in other words, what your experiences in your environment and the unique experiences that you occur over a lifetime, like that actually can change the way that your genes operate.
How freaking cool is that? Okay.
It's amazing. It's so cool.
Now, one more question, if I may, because I guess I'm trying to distinguish in my mind, and we'll give some examples, but like if a person, so we're talking about the influence, some sort of external influence, right? Something that's going on in our environment. Like, are we talking about things, sounds like we were talking about a lot of different things, but like nutrition, right? And I don't know, some of our behaviors, but that's somehow like help me distinguish epigenetics from like cellular changes or what happens if we're like smoking a cigarette. Like that's different. And how that might cause cancer, for example. Like how is that different? Can you help me understand?
Yeah, absolutely. Okay, so epigenetics, basically, the whole idea of epigenetics is that it states that we are not simply the physical embodiment of our genetic code.
Okay.
Right. And I think now might actually be a good time to talk about twins.
Yes.
Because that's actually, I think, to me at least, that is the most convincing evidence of epigenetics, is if you look at genetically identical twins or like monozygotic twins that originated from a single egg, they are going to They look exactly identical. But as anyone who has ever known a pair of genetically identical or monozygotic twins can attest, they are not identical human beings, right? They are going to have different personalities, different propensities, different, they can develop different diseases as they get older. And, I think is exactly exemplified what we're talking about by epigenetics, is that like if there were no epigenetics and we were just a product of our genes, genetically identical twins should be identical human beings, but we know that they're not.
Mm-hmm. Love it. Okay, so we're going to give some examples, some landmark studies. Obviously, Sarah, as you just said, there have been some really impactful twin studies that really help, I think, bring epigenetics to life. But I feel like there are a couple of terms that when I was reading through materials that I have no clue what they mean. So words like methylation, histones. Can you just set the stage? What are those things?
All right. Yes. Time for a little biology lesson. But don't worry. It won't be scary. Because
and Sarah, we're going to let our listeners into a little secret. We discussed the possibility of having a safe word. If Sarah goes too deep into the, biology, we didn't actually come up with a safe word, but I don't think we're going to need one. Come on, lay it on us.
Like coconut.
Coconut. All right. Got it.
Okay. So if I get to just, you just let me know, because I really think everyone will be able to get this. I think it's a beautiful concept. I mean, I'm so excited about epigenetics originated from a very simple and sort of elegant scientific question, which is are we a product of our genes or are we a product of our environment? And basically epigenetics states that it's not either or, it's actually definitively both. And let's maybe just try out this analogy with me.
We love an analogy.
I love an analogy. I mean, I think that's the best. It's just what makes biology so much fun. It's just bringing it to life with an analogy and just anthropomorphizing different aspects of scientific processes. I very much enjoy that. All right, so let's think of our DNA as a library full of books. And I love this analogy too because I love reading. A library full of books. Okay, and each book. So the library contains all the instructions for making a human being, and each book represents a different gene, and each book represents a different gene that codes for a specific trait. So that's height, hair color, eye color, attached earlobes, these traits that physically make up who we are. So we can consider epigenetic markers as bookmarks that are placed in specific parts of the book to sort of indicate which sections that we should be reading or focusing on. And also conversely, which sections that we should just ignore. So when we put a bookmark into the book, that's what is called for turning a gene on for expression, epigenetic activation of the gene. So that's when you put a bookmark in the book that highlights the part of the gene that should be expressed or turns on. And then the parts of the book that don't have bookmarks, those are the parts that can be sort of ignored and turned off or not expressed. Are you with me so far?
I'm with you. No coconut.
Okay.
Keep going. Yeah.
Okay. All right. So we're going to extend the analogy. Okay, so and those bookmarks, that's DNA methylation.
Okay.
So that's just, and what methylation is, I mean, methylation is just the insertion of a methyl group, and a methyl group is just a little chemical tag that is literally just one carbon atom and 3 hydrogen atoms. So it's wild to me that that small little chemical mark can just do so much. That just boggles my mind that that's a thing that happens. But this little insertion, this little bookmark, if you put it onto the DNA, that either activates or inactivates a part of that gene, turns it on or turns it off.
Got it. Okay. Now what about histones?
We're getting to histones. Okay. I love histones. I just wrote a paper that just got accepted for publication about histones.
Congratulations.
Thank you. Yeah, it's in Alzheimer's disease, but I digress. That's not what we're going to be talking about today. Okay. So, but what are histones, right? So histones, okay. We can think of histones like the covers of books. And if we think of a brand new hardcover book, that's kind of going to be harder to open, right? Because it's got a stiff cover. But if you think of a really soft cover, a soft cover book that you find in one of those little lending libraries on the side of the roads, we have those in Chicago. I think they probably have those everywhere.
We have those here in Western Mass. Yeah.
Right. An old Grisham novel, right? Yeah. It's just just easy to open and very accessible. The genes are going to be very accessible. So that's basically what we're talking about when we talk about histones. So histones, just from a biological perspective, they act as spools for the long, thin strands of our DNA to wrap around. So I mentioned we have like 3,000 million of these of the A, T, C's, and G's from our mom and 3,000 million from our dad. So it's a lot of material. It's about what it amounts to is if it were like all laid out, it's about 6 feet of DNA. And that needs to be packaged into these tiny little cells. And how are we going to do that? Well, we got to wrap them around. We got to make it compact. And that's what histones are. Histones are the proteins around which the DNA are wrapped so that it can fit nice and compactly inside of a cell.
Love it. Okay, now can you help us understand just in the context of epigenetics then? So where does the methylation and histones, where do they come into play? Is it, so you're saying like something externally is impacting the methylation and histones? And then that is then impacting gene expression? Or am I
correct?
Okay.
No, that's 100% right. But an important point too is that these epigenetic modifications can be regulated both externally, right, by our experiences in our environment, but also internally. And that's what's really cool about epigenetics as well, and what we've come to realize recently, is that epigenetics also plays a role in what's called cellular differentiation, right? So we have about 200 different types of cells in our bodies, right? Like not all cells are the same, right? Like the cells that make up my hair are different than the cells that are in my brain, are different than the cells that are in my heart. All of these different cells. And they all do different things. So how does a cell, but all of our cells in our body contain all of the genetic code, right? That's why, like, if you're getting a DNA test, you could do a cheek swab and it'll give you all of the DNA.
Okay.
But the cells in your cheek are not going to be expressing the same genes as the cells in your heart because they're not heart cells. Like, they don't need to. But it still has all of the genetic material in there.
Okay. I think I got it. So now, and tell me if I'm jumping ahead too much, but I'm trying to understand and maybe bring to life how some, let's say, I don't know, environmental factors could influence gene expression. Like, what are we talking about? You mentioned nutrition. That was just like one example. So like, yeah, diet, but also what about like stress? Just to I don't know, be transparent. I've often thought about epigenetics. I'm the granddaughter of Holocaust survivor. So I'm like, okay, could the trauma, that my grandparents experienced, could that have somehow impacted who I am? Even though, I don't know. I'm spitballing here, but what is epigenetics in the real world? Like, help bring this to life for us.
Absolutely. So I think what you alluded to was something called transgenerational epigenetic inheritance. And that's a little controversial, I will say, because a lot of the experiments that have suggested that this might be the case occur in very controlled laboratory settings using genetically identical laboratory mice. And this is very, it's very easy to manipulate certain conditions within controlled laboratory settings. So just to maybe give an example of like what you were talking about of this maybe transgenerational inherited trauma specifically, there was one study in laboratory mice that were trained to fear a very pleasant sort of fruity odor. Some mice have very large olfactory bulbs, so they're very sensitive to odors. Like that is their primary way of communicating with the world is through smell. So these mice were trained. They were given a sort of a mild, very mild electric shock. And basically what the experiment showed was that this fear of the odor was passed down to their children and grandchildren.
Wow, that's wild. That's so interesting. Okay, sorry, go on.
Oh, no. But that's a very controlled setting.
Right.
These mice were genetically identical and they were kept in cages under like fixed light-dark cycles, fixed diets. And But in the real world, when we're talking about humans, there are just so many different confounding factors. And I mean, like, Jess, you know this because you do all this kind of research with human subjects and population data, and you know that it's so different, right? When you're talking about humans than if you're talking about lab animals.
1,000%. And the tricky thing with humans is, we can't always control for all the confounders, right? We do our best to find proxies for variables and try to control for things using, statistical models, but it's imperfect, as you said. It's very, very different than a preclinical study in a lab done on genetically identical mice. So very, very good point. Okay. Did you, anything else to add there? Could we maybe talk, you already started talking about some of these studies, like what are considered landmark studies in epigenetics. So I guess maybe continuing the conversation about mice, there was the study on agouti mice. I love that. I love the word agouti. And I guess, so that's the type of mouse, right? They're, is that right? I think they're like yellow mice, right? And they harbor, and I'm reading a description of this. I might need you to help translate this for me. But so these yellow agouti mice, they harbor a transposable element in the agouti gene. And so let's see. So the researchers, and we'll link to this in the show notes. I believe the study authors were Waterland and Girdle. This was a study published in 2003. And so they manipulated the epigenome of mice by feeding the pregnant mothers different diets. And then that affected the coat color of their offspring and predisposition to diseases. And so just more specifically, the mice whose mothers were fed diets rich in methyl donors. So I guess this has something to do with the methylation that you were discussing previously.
Yep, 100%.
So we're talking about, I guess, folic acid is a methyl donor. So the mice whose mothers were fed diets rich in methyl donors had offspring with normal coat colors and lower risk of obesity and diabetes. And I guess that the takeaway was that diet during pregnancy could affect the epigenome of offspring and alter disease risk.
Yep, absolutely. And what's really cool about the agouti mice is that it gives you a very specific genotype to phenotype, like 1-to-1 ratio, because like this gene, specifically the agouti gene, when it's expressed or when it's turned on, right, when there are those bookmarks present, then the mice are yellow. And then if it's turned off, then or a lower level of expression, then the mice are brown.
So cool. Okay. So then moving on, and I'm probably going to mispronounce, is it Frega or Fraga? Do we know? I'm so sorry if I'm mispronouncing it, but there was a study published in 2005. It was the identical twin epigenetic study. And so this study examines epigenetic differences in identical twins over time. So it was a longitudinal study. The researchers looked at DNA methylation and histone modification patterns. And we now know what that means, thanks to you, Sarah, in pairs of twins, both young and old. And they found that while twins are epigenetically similar early in life, that older twins show significant differences in epigenetic markers. And these changes impact gene expression and help explain how different traits can arise from the same genetic background. And they were looking at things, by the way, some environmental factors like diet, lifestyle, and stress. So I thought that was really interesting. I don't know if you have anything to add about that or any comments on that study.
I mean, to me, that's the most convincing evidence for epigenetics, right? Is just the fact that we have two identical individuals genetically, but they're so different. Mm-hmm. And what's cool about this study is that there were multiple different time points that they looked at. Right. So so they looked at epigenetics in childhood, and like these young twins, they were pretty epigenetically similar. Mm-hmm. But then they looked at during older adulthood, and it's like vastly different epigenetic markers. And this, we think, is the reason why twins are different. It's just these epigenetic differences due to just different experiences in life, different environmental factors, different diets, different exercise regimes, like different interactions with different people. I mean, all of these things we're coming to learn can absolutely shape the way that our genes are expressed.
And I think this was one of the first longitudinal human twin studies, because some of the other landmark studies were done as we were discussing in mice and rats, in a preclinical lab setting. So this was very cool. Now, this next study, I'm curious. I know you have some thoughts on it, and I want to hear more. So it's the Dutch Famine birth cohort study. So I'll give a little bit of an overview, and then I want to hear. I want to hear your thoughts. But so this was a study that explored the long-term effects of prenatal exposure to famine. So this was during World War II, during the 1944 to 1945 Dutch hunger winter. And it was a study on epigenetic markers and health outcomes in offspring. And so the researchers found that individuals who were in utero during the famine exhibited changes in DNA methylation and increased susceptibility to chronic diseases like cardiovascular disease, diabetes, and obesity later in life. So tell us, Sarah, what is you have an issue with this?
I mean, it's not even so much of an issue. I mean, it is evidence,
yeah.
But I just, because I kind of alluded to this already, is like human lives are not as easily controlled as they are in lab studies in the type of work that I do. And it's like, we're not looking at genetically identical individuals. We're looking at genetically different individuals. And it's like, there are just so many different confounding factors that could have contributed to all of these differences that we're seeing in the study that it's really hard to pinpoint exactly, like, yes, this is due to epigenetics.
Got it. Oh, I love that. No, absolutely.
That's the only thing. Is that, like, I'm not saying like it's not the case,
right?
Or I disagree with the findings. It's just I take it with a grain of salt.
Mm-hmm, mm-hmm. I think what you're actually doing right now is really highlighting a larger conversation on research design and study design, and how there really are pros and cons to all of the different study designs, right? And so to your point, these very controlled lab studies, they obviously have their benefits because they are so controlled. We have so much control over the variables, which limits the introduction of bias and confounding and all that good stuff. But there are also downsides to that. I mean, obviously, we're also looking at mice and rats versus humans. So if we want to look at humans, then we're turning to different types of studies, and there's obviously an introduction of bias. So love that. There's one other study that I just wanted to highlight. This one I thought was really cool. This is the NASA twin study. I believe this was published in 2019. And so the study involved astronaut twins, so Scott and Mark Kelly. And Scott spent a year in space as an astronaut, whereas Mark stayed on Earth. And so basically, researchers just compared the effects of space travel on gene expression and epigenetics. And while the overall genetic makeup of the twins, of course, remain the same, they found that Scott's epigenetic markers, including, again, DNA methylation and gene expression, changed significantly during his time in space. And some of the changes reverted after his return to Earth, but others persisted, which suggests long-term impacts of space travel on epigenetic regulation. I just thought that was so cool.
That is cool. But like, again, and like my skepticism alarm is going off, because it's like, if you exposed twins to two different any two different experiences, whether that be like one of them climbs Mount Everest and the other one sits on the couch for a week, and like, or what I don't know, like one of them eats just blueberries every day, all day, every day, and then the other one just eats Cheetos. I mean, any sort of manipulation, like long-term manipulation like that, like, is absolutely going to have a differential effect. Mm-hmm. So it's just, it's difficult to say for certain, like that's also, it's an N of one, right? It's one set of twins, right?
Right.
And just going back to the Dutch famine study for a second, I would be much more likely to believe those results if all of the people in that study were genetically identical. I mean, which they aren't. I mean, people are not lab rats, but like, if they were, I'd be much more likely to believe the results.
Okay. Now, do you have any commentary on the role of epigenetics in mental health? Like, I'm just wondering, do we know anything, any connection to conditions like depression or anxiety? I mean, I know we touched briefly. It sounds like there's maybe some controversy in our understanding of, what did you say was intergenerational trauma?
Yes.
Do we have any understanding or any data studies on the role in mental health?
Yeah, there definitely are. We do know that epigenetics plays a role in a number of disease states, whether physical disease states or mental health. Conditions such as depression definitely do have a genetic basis, and there are certain genes that will predispose you to developing conditions such as like major depressive disorder or generalized anxiety disorder. But then there are certain experiences that people can have across their lifetime that would sort of kick those genes into gear, or like epigenetically activate them in ways that are definitely, it can definitely not just be attributed to the genetic code itself.
Got it. Okay, that makes sense to me, because I guess that makes total sense, right? Because there is likely a genetic, not likely, we know that there's a genetic component to the development of mental health. And so we're talking about genetic expression. So why would that not impact mental health?
Right. But it's not a 1-to-1 ratio. Like we haven't identified like the depression gene where like, if you have that gene, you will develop depression. So that I think is definitely, can definitely be attributed to epigenetics. And there are other disease states too, such as certain cancers that we know are epigenetically regulated as well. There's a certain, so when a tumor suppressing gene is epigenetically silenced, we know that this can contribute to the spread of cancer cells or the tumor progression or resistance to cancer treatments. So that's, more of like the physical side of the physiological disease rather than a psychological disease. Right. We also know that there's a role of epigenetics in heart disease. And the modifications that are linked to, like plaque buildup and can lead to heart attacks and stroke. And this is why, like some sets of identical twins, like one of them can develop heart disease while the other one doesn't, because heart disease is definitely influenced by a number of lifestyle factors.
Mm-hmm. Got it. Thank you.
Yeah, there's all these things. I mean, there's also, and then like my area of research, right, is just Alzheimer's disease. I mean, there is evidence to support the epigenetic regulation of certain genes in the regulation of conditions like Alzheimer's disease or other types of dementia.
All right, Sarah, I think now's a good time for our Heard from the Herd segment. So I opened up a question box. Yes, I opened up a question box on our Instagram, which I do before we record our episodes. So definitely follow along on Instagram at Unbiased SciPod, and you will have a chance to drop your questions. I love this one person wrote, so I said, drop your questions about epigenetics. And this person wrote, what the heck is it? But I think we covered that. I love that. Thank you. All right. So the next question is, can we actively have an impact? They said on them. I think they're talking about on our genetic expression. Like, what do we do with this knowledge? Like, can we or should we be doing certain things? I mean, it seems like not an easy question to answer, but how would you respond to that?
Not at all. Yeah. Because we're just starting to scratch the surface. Epigenetics, the study of epigenetics is actually relatively new in biology. Yep. We have not known about this for very long at all.
Yep.
So, we're just starting to scratch the surface about like how we might be able to like influence, like how we might be able to put this information into practice.
Mm-hmm. Mm-hmm.
I would just say that from the limited data that we do have, things that are good for your overall health are going to be good for, epigenetic regulation. And they do affect your gene expression. Like there are studies on exercise, there are studies in nutrition. Like we know that these things can definitely have a great impact over your genetics. Like even if you have a genetic predisposition for, say, obesity or heart disease, if you implement certain changes in your lifestyle, you are not just a product of your genes. Like you can actually make changes.
Got it. All right. Then we got a set of questions, and I'm going to do my best to sort of, like, there was a theme in them. So I'm going to try to truncate to one question, but questions about now, advancements in technology, and like what comes next. So people were asking about things like CRISPR-based tools, next generation sequencing. Someone asked about single cell epigenomics. I have no clue what this means. But basically, like, with this knowledge, are there ways, are we on the cutting edge of figuring out ways to actually manipulate our, epigenetic changes in specific cells and tissues?
We're on our way. I do think we're kind of far off from being able to manipulate the epigenome because it's so complicated. There's so many different factors contributing to it. And you mentioned CRISPR. CRISPR is actually a gene editing tool. So CRISPR operates at the level of the genes, and not at the epigenetic level.
Got it.
With regards to single cell epigenomics, I actually use this in my research. I didn't think we were going to be talking about this, but basically what that is just looking at one type of cell. So in my case, that's neurons, cells in the brain. I'm a neurobiologist. And what you can do is kind of similar way to how, we mapped the human genome. You can map an epigenetic marker across the entire genome and then look at different levels of this marker on specific genes. And then from there, you can sort of determine whether or not you can deduce whether or not that gene is being actively expressed.
Got it. Okay, so some very cool applications. Potentially, we're not quite there yet. Okay.
Yeah.
And then similarly, I got a lot of questions about personalized medicine. It sounds, I would imagine you're going to have a similar answer. I don't know if you have anything else to add just about personalized medicines, but yeah, I don't know if you could see Sarah's face right now. She's crinkling her, crunching her forehead.
I have, for those who aren't listening, for those who aren't watching on YouTube, I got a lot of facial expressions. I can't play poker. I don't have a good poker face. Maybe. Okay. Oftentimes in science, like, I wish that I could just go into hibernation for about 100 years and then wake back up 100 years from now when the technology is more advanced and then keep doing my science, because the technology it's rapidly progressing, but it's just not quite there yet. We maybe at some point will be able to use epigenetic markers to predict specific diseases, but I think we're more, I think right now we're just starting to get to the point where we're able to identify specific genes that map or increase your risk to specific diseases. And, now to layer on top of that, the epigenetic regulation of those genes,
right.
There's so much going on.
Yeah. And then I guess the last set of questions that I came across, people were asking about bioethical concerns and what if our understanding of epigenetics translates to a technology that's used for bad. And I mean, I don't know, my gut reaction is I don't even think we're close to that right now. We're still trying to understand more about the epigenome and epigenetics. And I think right now, how wonderful would it be if we were able to, and this is my non-neurobiologist, layperson, turn off genes that will, increase our risk of cancer. I mean, that's where I think we're going right now. I mean, could it be used for bad? I think we could say that about any technology, right? But for me, right. I mean, I'm just so excited about this. I mean, it's incredible, and it's so funny. I actually remember when I was doing my NPH, and in my early epidemiology classes, this was 15 years ago, we were it was this epigenetics, it was this so cool. But that's really where our knowledge stopped. It was there's this cool frontier, new frontier in science, and we're really understanding. I feel it's a cool link, actually, Sarah, between my world and your world. I'm more epi and you're the, the biology, the more biological level. It's so cool. And so I think it just drives home. And I'm sorry, I'm going to make just a quick, data science reference. When we're building models like regression models, logistic models, we add variables. And the whole thing is adding in variables that we think are having an impact on an outcome. And so like my thinking about this is we'll add in some of these, I'll just very simply say like these external factors, more environmental factors. And then we try to include things that are proxies for genetics, right? And our best way to do that, at least in my world, is by, like race, ethnicity, things like that. But then we'll also have interaction variables. So you'll put these things in individually, but then you'll also have interaction variables. And for me, like, that's just like, I don't know, that is very relevant to our discussion here. There are these external factors, these internal factors, these genetic factors, and they have their individual influence. And then obviously there's something going on, this intermingling, and that intermingling is also obviously then impacting outcomes that translate to health and all these other things that we're discussing. It's so cool.
Yeah, and I think you bring up a really important point too about epigenetics, is that we've been talking a lot about like the external effects on epigenetics, but there are also internal effects on epigenetics. And I briefly mentioned cell differentiation, and I think the best example of that, and I do give this example in the Alzheimer's disease episode, but I just think it's the best one, is that like when we talk, when there are different cell types in our bodies, and they all have every single one of these chemical base pairs, these A's, T's, C's, and G's, they're in every single one of our cells. But every cell is not created equal, right? So the cells in your stomach are not the same as the cells in your eyes. And there's a certain gene in your stomach that produces this gastric acid that makes it a very acidic environment to help you digest your food. We have that same gene in our eyes, but it's not expressed. Our eyes are not producing hydrochloric acid. Like, that would be a really big problem if they were. But that's epigenetics at work, right? We have the genes in our eyes and in our stomachs. They're the same genes, but they're not all being expressed. And that's the internal side of epigenetics as well, which is just as important and also a little bit easier to study.
That was a great example. Sarah, do you have any last thoughts or takeaways for folks? I mean, I'm walking away from this optimistic and excited, about this field and how it seems like we're learning more at a rapid pace. But any final takeaways or thoughts, things you think that we didn't mention that we should? I feel like we covered a lot of ground though. So
we really did. Yeah. And I mean, I the one thing that I hope that people got from this episode is that it's not nature versus nurture. Like, the whole idea of epigenetics is like, it is nature and nurture, sort of definitively. It is the bridging of the gap between your genetic code and your environment. And it just pretty much dictates that you are not a product of your genes. There are things that you can do to change their expression. Who you are is not baked into your DNA at birth.
I want to give a shout out to you, to my amazing co-host, Sarah, who took this concept, this topic is so complex, and you really, really made it digestible for me. I might have shied away from this topic previously, but you really brought it to life. And really, for those who, I know we're just getting to know Sarah, but the thing that I love most about her is really her passion for science communication and taking these really complex concepts and just helping us all understand them. So thank you, Sarah. Really, that was an incredible discussion.
It's my pleasure.
All right, everyone. Well, thank you for tuning in to Unbiased Science. If you haven't already, please be sure to subscribe to our Substack. That's our weekly newsletter. And Sarah is getting more and more involved in co-authoring those newsletters, and they're incredible. We're able to really comprehensively tackle topics from both a biological perspective as well as public health and population health perspective. You can sign up at theunbiasedscipod.substack.com. It's totally free, although there is a paid option if you're able and willing to support our science communication efforts. Let's see what else. Leave us a review wherever you're tuning in. Apple, I think you could do so on Spotify, but definitely on Apple. That helps us. It helps us with visibility because obviously we're doing this because we want people to tune in. So definitely leave those reviews, please. Also be sure to subscribe on YouTube. Yes, we record video, so we get out of our pajamas and get at least partially dressed so that we could record this so you could watch on video. Thank you all so much for your support, and
thanks for tuning in for No Nonsense, Just Science.